Magandang umaga po sa inyong lahat. Isang kagalakang muli na tayo magkita-kita dito sa Kwentong Likas Yaman. Katulad ng dati, lagi kaming merong bagong sorpresa at bagong pag-uusapan. Pero bago yon, pwede ba? Lagi naman kaming pinapanood nyo. Mag-like naman kayo or mag-subscribe. Alam ko marami ang nakikinabang dito, lalo na yung mga teachers na ginagamit yung session namin para mas mapaliwanagan ng ilang mga kaisipan sa mga nakakonektang mga topic na pinag-uusapan nila sa klase. I know na may mga na-meet na ako, lalo na mga graduate school and college teachers na nakakonect sa lectures nila yung ibang sessions ng Kwentong Likas Yaman. So, konting hiling galing sa amin. I-like nyo ha! Ayun, ako si Doc Letty at ang aking katuwang na si Doc JD ang magpapakilala ng ating panauhin sa umagang ito. JD.
Salamat, Doc Letty. At ngayong umaga na ating episode ng Kwentong Likas Yaman ay ating ipapakilala ang isang balik scientist, no? And I just want to mention, gusto kong banggitin na nagkita kami sa isang balik scientist convention, sometime I think this October, no? October ba 'yon parang doon ko siya na-meet. And then I mentioned to him na we have a blog and that we have this Kwentong Likas Yaman at kung pwede siyang mag-share ng kaniyang research, no, sa ating programang Kwentong Likas Yaman. His name is Dr. Romel Santos. Napakabata pa niyang tingnan lang. But he's not that really that young, di ba?
Huwag mo na i-reveal.
Looking lang siya. Looking talaga. But of course, nasa calendar pa ba? Di ba? Nasa so, ibig sabihin bata pa. But anyway, he is a graduate in Bachelor of Science in Neurobiology. Yeah, tama, neurobiology, and his PhD in biological sciences na kinuha niya pareho sa University of California, Irvine. Okay? And then for some luck, no, siguro sa swerte, no, nag-apply siya sa balik scientist program ng DOST, and luckily he is now connected at the Institute of Biology at UP Diliman. And today, isi-share niya sa atin ang kaniyang research, no, on the topic developmental biology. So, magbati ka and you greet our listeners in this program, Kwentong Likas Yaman, Dr. Romel.
Hello. So, hi. Thanks for the introduction. And really, I'm very honored to be taking a part of this podcast and / discussion. So I'm ready to have some fun and friendly discussion related to developmental biology. So thank you again. So yeah.
Okay. So for the first question, I would want to ask Dr. Romel, kindly explain to our listeners, ano ba ang developmental biology as a branch of science?
Yeah, so for the term developmental biology, it's a pretty umbrella term actually. But essentially, it's studying the development of some sort of animal. And this sort of development is composed of many events. It could be like the sperm fertilizing the egg, and the zygote, which you call or fertilized egg, is actually dividing. So those events are what compose like developmental biology. And eventually, yes, this fertilized egg is dividing and eventually forms like these 3 germ layers called like ectoderm, mesoderm, or endoderm. And those give rise to eventually like our limbs, our skeletal muscles, our bones, our nervous system. And so, yeah, again, I'm saying that developmental biology is such a very umbrella term just because it can be narrowed down into specific developmental processes. Like in my case in graduate school, I was focused on developmental neurobiology. And so that's the study of how the brain forms within the developing animal. And even that in itself has like very specific branches. Like my focus in grad school was just focusing on the development of the visual system of the nervous system. So you can really see how like specific the field can get, but how broad itself it is, like if you take a step back. But that's this exciting thing about it. Like there's so many research that's going on within this developmental biology field that even if I'm in the field of like neuroscience, like I'm still keeping up with other fields. Like stem cells is another related form of developmental biology, where again, that's the idea of like how these pluripotent stem cells or have the ability to differentiate into like these either mesoderm or ectoderm or like just specific cell types that kind of make a nervous system. So it's very broad. And yeah, I know it's just like a very like what's developmental biology, but yeah, it's really encompassing a lot of things. That's exciting.
Yeah, having done a lot of studies in here and having gotten a lot of exposure, I would like to ask a little about the history, because when I took this course up in UP Diliman, it was not developmental biology, it was embryology. And then when I started teaching it a few years later, and for something like 10 years, it still remained as embryology. But maybe 5 years much later, it became developmental biology. What could have been the reason for that?
Yeah,
Would you know?
I was reading a paper based on that. Like the classical term of is called embryology, but like the modern term nowadays is technically developmental biology. I think the reason being is like embryology, it sort of encompasses like the starting point is like the fertilization of the sperm on the egg. And then all the way to eventually, okay, 3 germ layers, then eventually like the limbs of the growing embryo. And so that in itself, from, I guess this is like my understanding, like what encompasses embryology. But then to encompass the other fields, now we're really talking about like the broader scope of embryology. I think that's how it transitions now into like developmental biology. More than just like embryology events. It's more like, okay, what about regeneration, or again, like the stem cell field?
Oh, okay.
Yeah. It became more encompassing.
Yeah. Like injury and repair type of. And so that's what
it became a bigger umbrella. Okay, because embryology would be the study of the embryos.
Early stage. Okay.
So how did you get into these interests? There are very few. I know that there is are you aware of the Developmental Biology as organization or association in the Philippines? The head of that, the president is Ryan De Chavez.
Oh, Philippines? Oh, yeah. I think I'm familiar with some of those organizations, but I know there's this Filipino Society for Developmental Biology.
That's it. That's it.
I think.
That's it. Actually, the current president is our division head right now. That's Ryan. Yeah.
That's exciting. Yeah, I think there's a convention 2 weeks from now, and I'm going to attend. Maybe I might be able to see you over there at.
No, we won't. I'll be in biodiversity. That will be 2 weeks from now also, first week of December.
Okay.
Yeah, because usually it's being done. I already attended at least around 2 of their conferences ng developmental biology in the Philippines Society. I think it's a society. Yes, it's usually at this time. No?
It's in November, so yeah, I'm excited for that. There's a promising talks during that event. So.
Yeah, the first one, the whole of the first convention on that, my whole department was involved. We were the support group of that convention. Since it was just the first one, it was not thoroughly, extravagant, something like that. No, it's simple, straightforward, but still rich in information, and it was effective enough in putting together the people involved in studies on developmental biology. I think this one is going to be the 3rd one.
Oh, I see. Pretty exciting.
So going back to our topic, developmental biology, may I know the specimens that you're using in studying you were saying you are involved now in the neurobiology or sort of brain, something like that.
Yeah, so the thing about developmental biology is that we tend to use animal models to study these developmental events. And one of the advantages to these animal models is that you can pull out cellular mechanisms or study molecular signaling pathways from these animal models. And that's the advantages to using those, animals in the lab. But in grad school, I used the amphibian model, so tadpoles pretty much. And the thing is, the advantage to using that is that when you sort of get the fertilized egg, those eggs develop externally from the mother or from the female frog, compared to us humans, where like the development is contained inside our in the female stomach in this case. But of course, I'm kind of like becoming very technical. But the idea is like, yeah, I'm using amphibian models in grad school, but now here at in the Institute of Biology, I'm transitioning to zebrafish work, and we're using that as an animal model to study developmental events. With my interest is like how the brain develops in those zebrafish models.
Mhm.
Yeah.
So, but you mentioned about how do you make that see the image of this development in the brain? Say, for
Yeah,
example,
so
a
it's
tool. Are
actually
you
show
using?
you some examples if you don't
Yeah.
mind if I'm sharing my screen. So
Okay.
Okay.
prepared some
Please do.
slides to kind of give you an idea of like how actually we're using these animal models, but in combination with these fluorescent imaging tools. And so you can see here, this is the head of a tadpole that we fixed, but then we're able to label like specific tissues with antibodies. And those antibodies will eventually be stained with a fluorescent marker. So you can see here, like the green is like a green fluorescent marker, and the red is also like this red fluorescent marker. And so again, like this is the sort of the power of using like an animal model, just because you can take something as simple like a tadpole and stain it with like markers that you're interested in. So in my case, I was interested in like the brain tissue. And so you can see all these nerves scattered throughout the brain, and also stain for specific protein expression within like these brain tissues. So this is what you're seeing in red. It was a protein expression, but then just to see the outline of like the nerves, you can see in green. And so, yeah, this is just a combination of using, again, like immunohistochemistry, the, embryology of like the tadpole, and then also with microscopes that are able to detect these markers. So then you can get like a nice, really interesting image to study, and gives you a lot of data about like the structural like formation of, let's say for this, the CNS in this case. And so
I'm curious, at what magnification do you make these observations? Like,
actually, yeah, so the thing, that's a very, I don't understand the question, but like it's a technical method. So this thing is like, the thing about this microscope that we're able to do is that I can zoom in at the 63x magnification. And so like this part right here would be like a 63. And then the cool thing about this microscope is that it can take like maybe like 5 scans, like here, 2, 3, 4, 5, and like another row of 5 images, and then stitch it all together so that it becomes this whole image. So it's like of course, I'm shooting it at the 63x magnification so that I can see a higher cellular resolution. But then if I want to see the whole animal, then I have to stitch together those images to form like this bigger image. And so that's part of one of the cool things about the microscope that I was using at, I took it at UC Irvine, and they're able to kind of compile all this images to form this bigger image. And so, yeah.
Okay, so I mean, I'm just curious also with the kind of stain. Are you using stains, or I'm not particular with how fluorescent microscopy work, so
Yeah.
asking that.
Yeah, so this is the term, fluorescent microscopy is like the idea of to be able to visualize those fluorescent markers, but in order to specifically stain like a portion of a tissue. So what's in green is actually, I might get too technical here, it's like filaments. Like, so we're familiar with filaments are like embedded within a cell, but in the case of the optic nerves, there's also filaments like sort of contained within the nerve and these tissues. And so there's these antibodies that specifically bind to those filaments.
Wait, would you mean this would be some kind of the dendrites or the axons, or
Yeah,
it's a
exactly.
different thing?
Yeah, this is actually axons. So yeah, it's like filaments
Okay.
that bind to the neurofilaments of axons in this case. So
Mhm.
that's a good point that you sort of brought up. So these filaments, like yeah, that's embedded within the axons, but then you have like this primary antibody that
Mhm.
specifically recognizes a filament. But then on top of that primary antibody, which is binding to the filament, now we have a secondary antibody that binds to the primary, and the secondary antibody is what we call the fluorescent molecule. And so in this case, we call it like a 488 molecule that likes to bind to the primary. And so then a 48, I know this is more technical in terms of physics, but when you have like a 488 molecule or fluorescent molecule, you can hit it with a laser. And so when you excite that fluorescent molecule, now it emits like a light signal. And so in this case, to kind of simplify it, the signal that's being emitted is like this green light that I'm seeing here, or what this image is being captured. And so then, yeah, of course, if you kind of think about it, like take a step back and understand, like this is all the filaments that are being stained with the primary antibody, then the primary antibody is being detected by the secondary antibody, that's the 488. And then with the fluorescent microscope, we're hitting it with like some sort of laser light to excite that 488 molecule to emit that light you're seeing here. So yeah, this is why I kind of love developmental biology, just because there's like some art to it that like you can really see like the structural formations of like, let's say the axons that are innervating the brain. It's something that you don't really see on a day-to-day basis. Yeah, that's an exciting time when I had captured this image here.
For that particular specimen, what's the age of the which is the animal model?
So for this, it's like I think 5 days after fertilization of the egg. So yeah, so I think the germ layers are beginning to form like after 3 days, but then you start to get like the central nervous system beginning to form along just around that time period. And so then you have like the, again, what I was interested in, it was like the visual system. And so you can see the eyes and how it sends like axon projections, what we call the optic nerve across.
That's sagittal, right? I mean, from so what is the section? I'm just
so this is more of a top to bottom perspective. But one of the cool things about this staining was that it's the whole embryo. I didn't cut it up. It's what we call whole mount.
Yeah, whole mount.
And we didn't cut anything. We sort of just treated it with reagents to allow the the markers like the 488 antibody to penetrate the tissue, and then we're able to get the final result here, like this whole structure being labeled.
Is this the frog or the tadpole or a fish?
This is the tadpole,
okay.
Just the head. Yeah, you can't really see the
head portion.
Yeah, the head portion. So if I can go to next, technically this is what I'm showing you here, just like this of the tadpole. Sorry if it's like, oh, it didn't look like anything, but now you can see like, oh, the tadpole head. But yeah, this is like one of the beauties of an animal model system. Like you can study the organism, but then really go down to the cellular level. And then in the case of like neuroscience, which is like the synapse is also a focus of interest, you can also begin to study like, down to the level of the synapse. And that's just a very valuable information for us, I guess, in the neuroscience community. Like, how do these, like you're saying, like dendrites and axons, how do they communicate with one another? It's these like synaptic connections. So.
But you're using a different microscope. I mean, can you see this using the fluorescent microscopy in the synapse? Is that possible?
I think the resolution, you'll just be able to see like dots. I actually don't have any images of that, but there is a protein marker that labels like, let's say a synapse. And then let's say with a 488 or more of a protein, you can use a green fluorescent protein to just mark a synapse. But then how it appears like in the, I guess when you visualize it, it's just like a dot. And so yeah, I wish I had some images to show you of like of a synapse, but
yeah, let me ask something. With those studies that you have been doing, what were basically your and target results. What knowledge did you want to bring out? What kind of understanding has been revealed because of these types of studies so far? And what do you intend to do further on?
Yeah, so this is really based on the work that I did in grad school, which is sort of inspiring to me to what I'm doing now at UP de La Man. But one of the overall questions of what I was working on as a grad student was trying to understand these molecular guidance cues that shaped these neuronal tissues, or the way, like to be more specific, like circuits within the brain. So remember, there's all these neurons within our nervous system or nervous tissue, and like axons are connecting to dendrites. And so what we're interested in was understanding how, let's say, this cell surface protein was directing the formation of those circuits. And so generally, in order to understand that question, we did like a knockout approach where we introduced this knockout reagent where we'll downregulate the expression of this protein. I'm going to call, just for now, to kind of give it a name, like this DS-CAM protein. And so we introduce like some sort of reagent to knock down the expression of that protein. And so we're able to deliver it across just half of the embryo. And then you can see that, yeah, there's this downregulation of the protein, so that you don't see the, relative to the control side that doesn't have the reagent that knocks down the protein, you can see that the DS-CAM protein itself is like lower in levels, intensity. So you can see that there's this, like a shadow here that compared to the control side. But yeah, here's the main point that now we're able to use that reagent to introduce it into, let's say, a single cell and see how is that affecting the morphology of those neurons as a result of downregulating that protein. And so, yeah, this was like a finding. One of my first papers in grad school was based off this work that's showing that a protein called a Down syndrome cell adhesion molecule was actually important in guiding the formation of dendrites within these neurons in the visual system. And so, yeah, that was a lot of just understanding the structural anatomy of just like a single neuron. But then you can also complement this structural data with functional data. So we know that the visual system is involved in our perception. And so one of the things that I did, so yeah, I'm showing that there's this knockdown of the protein, but we can also deliver it throughout the visual system and see how now the animal's reacting to a stimulus that it tends to react towards. So you can see that this tadpole is swimming in this petri dish, and typically it will perceive a stimulus and react to it, and it will like usually swim away. And the idea is that, okay, let's sort of manipulate the structural integrity of the visual system by, again, down-regulating that protein. And so what happens is that now there's this, disruption in the formation of those circuits during development. And so what we saw with the data was that these animals do not react as much to the stimulus compared to normal, functioning brains of tadpoles. And so that was just a very elegant work. So this was helped, but of course, like, yeah, I had assistants with me to help me gather this data, and but it's all published in my first author paper at the time. And so, yeah, this is what was done in grad school. But now, as I'm sort of, as a biological scientist at the UP de La Man at the Institute of Biology, I'm sort of transitioning now into the zebrafish model. And the idea is like, instead of doing like at a single cell resolution or single cell manipulation, or just like this just tissue manipulation, what I'm hoping is that to be able to introduce a transgenic technique where now I'm altering the genome of the entire embryo. And so we're really starting from scratch here. And yeah, this is what I hope to accomplish as a biological scientist, to be able to sort of introduce this transgenic technique, which is kind of similar to what I was doing here, introducing like these manipulations at the single cell level, but now I'm trying to do it at the more whole embryo level. And this is like, for example, one of the starting projects I'm trying to do is like introduce a green fluorescent protein that tags the cardiovascular system of the developing zebrafish. And it's just a starting point. Like, I know that it's not novel, like a lot of labs have already created this a cardiovascular model within the zebrafish. But what I'm hoping is that once I've sort of troubleshoot this technique at the Institute of Biology, we'll be able to now kind of experiment with other animal model systems, like the giant clam. I know someone's that's working with at the Marine Science Institute, or there's also someone working with sea corals. And I hope to sort of play around with these techniques so that we can also, visualize cardiovascular development in the sea coral or the giant clam, if that's even possible. It's just, this is just so far the starting point where I'm at.
Yeah, let me ask a question. What would be the practical usefulness of these results? Like, for example, being able to refine the method of injecting and, weakening the impact of certain proteins or stopping their effects. How will it be practically useful?
Yeah, so I've been thinking about that question very deeply, just because I'm trying to write a grant, and you see those grants are always asking those sorts of questions. And so really, I think my way of thinking is more towards like this climate change battle that we're sort of facing in the country. And so one of the things that I hope to do, especially with the transgenic labeling, is that, let's say with these either sea corals or the giant clams, that we can at least, before climate change, takes its toll on those population of animals and sort of wipes them out from the face of Earth, whenever, it doesn't, I know I'm probably over exaggerating that, but at least then, we're using these transgenic tools to at least catalog the development of those species, like the giant clam or the sea coral. And so that we'll be able to have like a database of information, like, oh, so what was actually the development of those species, just in case, like, if they ever go extinct. But especially for like, it's relevant for sea corals because those are the very, how do I say, vulnerable species that are being affected by climate change. And so what I'm hoping is that I'm able to catalog those development of like the sea corals, and so that we have a database to always reference, just in case, these sea coral species are wiped out, and we can always reference like, okay,
so you would mean that once you have cataloged this and monitored this step-by-step procedures of development, if you have, like, for example, you have cryophilized portions of these corals and kept them somewhere out there, you can regenerate them using those procedures, something like that, to replace the original ones.
Yeah, that if we could, I think the whole culturing of sea corals, that'd be that'd be the way to sort of like, oh, if these species get wiped out. But I never had thought of it that way. I was more like, let's just database these animals, and if we ever needed to reference them, because it's specifically like developmental, like monitoring the events during development, it's very hard. It's not a lot of labs or facilities have those tools to create those time-lapse images. And so it'd just be nice to have those images kind of reference in case like we want to do a comparative study across different species. But yeah, I think a lot of labs in terms of that, that's how they're trying to fight climate change. Like, okay, can we salvage these animals so in case that they're wiped out, they can bring them back into the population. And so those are pretty very
honestly, what I was thinking earlier when you were presenting those fluorescent images with the eye and the optic nerve there, I was thinking, if you are able to document the processes and the step-by-step conditions of development, I was thinking that one practical thing is to be able to understand ways by which you prevent pre-born or born mutations. Like we have we have Down syndrome or eye visual diseases that is inborn with a child or an animal, a big whale. but you detect that there is an abnormality inside the developmental process, you can kind of intervene to put in whatever the protein that causes that damage can be slowed down. It
Yeah, like
can be
CRISPR. Yeah.
immobilized so that the development becomes, it becomes normal. So instead of having an offspring that is abnormal, the abnormality is prevented right at the embryological stage. That was what I was trying to imagine earlier, no?
Yeah, that's like that's a great idea. I know like several labs are actually doing that with CRISPR. I know there's this controversy in China too that someone actually tried to do CRISPR on a newborn. So it's not really ready yet for humans, but I know, yeah, like a rescue experiments are being performed also like on like mutant models. And so those are also like very practical experiments. Especially like if you want to focus on diseases that are prevalent here in the Philippines. Yeah, that's like very great idea.
Practical and useful, maybe, for especially for human health. We tend to do that. But there are also high risk organisms that we need to save, like for sure the whales, the cetaceans in the seas are highly vulnerable and very much subjected to a lot of stresses like bomb testings, pollution, and many of those things. So if we could understand their developmental processes, maybe there's a way to protect them so that they would have less abnormal or mutant offsprings like that. And these are the high level and most vulnerable, among the most vulnerable organisms that we have. Well,
and I think since he is into developing database, for this experiment, so I think that will be very useful. Sometimes you do not know what will be happening in the future with all of this. Remember that now we are into big data. On the other hand, but I think having this database, for this particular experiments, I think that will be very useful. And sometimes you still do not know what will really be the exact or specific usefulness of those, diba? But in the future, who knows, it will really be very significant, and maybe
those are like the ideas though. Those are, I'm still trying to write that grant, so many good ideas there. So I'll talk about with you. I'll kind of follow up and message you about it. Like, oh, hey, can we?
Yes.
Work on the cetaceans. They are very vulnerable. I'm a wildlife biologist. That's why I have that concern. We have a lot of wildlife species, both in the marine and in the terrestrial environment, that have always been subject to a lot of threats. That's why we have a red list of organisms already. I'm part of the red list committee for the Philippines. How can we avoid them from getting extinct? That's what we would like to work on. Aside from the threats of climate change, we have subjected them to a lot more. So if we could minimize the number of possible mutations that can occur within the system by understanding the developmental process and intervening where mutations can possibly occur, then we give them a little ray of hope. Something like that. So work on the wildlife.
Yes.
For your next project.
Yeah. Thanks for those ideas. Really, those are, of course, like, I'm gonna reach out to you at some point. Like, oh, remember what you said during this discussion? So then let's write a grant together or something like that. So yeah.
So I'm curious about the Balik Scientist program that you are in. What category? I mean, in what particular kind of program? Because I understand that Balik Scientist program has different. In terms of whether it's short term, long term. Can you?
Yeah. So I'm under the Department of PCHRD. So that's more like health related. And so I'm currently doing the long-term engagement, the 3 years. And like the reasoning being is that, I really felt like the projects that I'm proposing especially like for the transgenic, it takes like time, and I wanted enough time to be able to at least like optimize these techniques and like be able to like produce an output. And so yeah, that's really my justification for doing 3 years. And yeah, just because I'm studying like nervous system development, that's also sort of health related. So that's why, yeah, just naturally, okay, like I'm gonna shoot for a PCHRD. But the thing is, like, DOST is pretty accommodating. Like at first, like I had an application and I was like, okay, like see where it sort of fits within like your I guess priority aims, and they're able to work with me. And like the final like I think evaluation, they said that, oh, PCHRD seems pretty relevant. And so then now that's where I am. I was able to get like a submitted application like I think last year, and they're able to accept it. So yeah. Mhm.
I
Sure.
have another question, not on the Balik Scientist program. What
Mhm.
do you mean by transgenic? You know, it rings a bell to me. There are usages to that, but for this particular study of yours, because if you say transgenic, it has to be transcending like species, like it is, you know, it is found in every other species or genera, something like that, or it might be a gene that is found but I really would like to clarify
Yeah,
that.
so I think it's to kind of define it simply, like transgenic is more like to transfer genes. I think that's another way to kind of describe it. And so in terms of like this transgenic technique that I'm performing, like this is an example. So this is like a fertilized egg that's already at a 1-cell stage. And so you're able to introduce like this foreign gene into this developing embryo. And so it's at the 1-cell stage. And so this is like literally the first stages of like development. And so like, let's say I'm inserting this green fluorescent protein, or like a gene that will express a green fluorescent protein into this well, technically this is the zygote, but it'll eventually transfer over to the 1-cell stage, which is, you know, this is the beginning, you know, or beginning of beginnings of the embryo. And so this 1-cell will incorporate that sequence that will begin to express the gene fluorescent or green fluorescent protein. And so then as this begins to divide, like, you know, 4-cell stage, or then it's like this whole sphere of like cells. So this gene is actually being passed on to each of these individual cells, and eventually, again, it's that formation of the 3 germ layers, right? The mesoderm, ectoderm, endoderm. Eventually, you form like these, you know, prominent structures of the embryo. But again, like as it's dividing, all those cells are uptaking, or at least passing on the gene that I first introduced at the very beginning at the 1-cell stage. So then all of the, you know, subsequent cells will eventually express the green fluorescent protein. So, and the thing is, like, I know this is another technical thing, but there's the green fluorescent protein, but you can control like what cell types will express that fluorescent protein. And so in this case, like there's like something like KLDR, which is usually a promoter that's upstream of your gene of interest that you want to express. And KLDR is something that you'll mostly express. You might have to correct me if look at this up later just to kind of like fact check, but it's usually like in cardiovascular cells that will begin to express it. So that's why not the whole embryo will express it, it's just like specifically tissues expressing it. So yeah, that's the whole idea. I know the whole question is first what's transgenic, but yeah, that's like the movement of like genes into like the genome of the animal. But that's my understanding of like transgenics.
Okay, follow-up question. Sorry, because the term, you explained it well anyway, but it stirs my imagination and understanding. When you are the materials or the basically you're using tadpoles, no, then there is an assurance that this has been completely barcoded, so you know the complete genetic sequence of the chromosomal compositions there, and then you insert a plasmid into that whole system of DNA codes. Isn't that disrupting the whole genetic process of the organism?
I think you can create some sort of mutations.
Yeah.
Induced.
Yeah, like induce like a mutation and everything.
And that's the thing about transgenic, right? You are transferring a gene from related or even a non-related organism.
The
That's
sequence,
how I
yeah.
understand it, diba? You manipulate more or less
Yeah,
the
so yeah, things like I understand. Yeah, these are like standard techniques, and like a lot of labs are sort of like introducing genes. But if
Okay.
it were to create like a serious or induce a serious mutation, the like wherever like the random place like the gene of interest is being inserted in, um, the animal wouldn't really develop properly. So then those you don't really use those. But I guess in the cases where it does insert into like a place where it's like viable and you get like a sort of a normal embryo, those are what we work with. But yeah, I understand. Like even I kind of think about those whole process of like, oh, what if I insert this like okay, like the green fluorescent protein? Aren't I creating like like a point mutation or like
Hmm.
a translocation of a of like
Yeah.
chromosome? But of course, like yeah, if it was very detrimental, it would have these animals wouldn't really survive. And so I guess the interesting thing to note is that really these like techniques, there's really a 10% success rate. So let's say you do 100 embryos that you're injecting, like only 10 will actually kind of like manifest like what you're sort of interested in. But yeah, it could be the reason like you're explaining that you're inducing like maybe these detrimental mutations that
These three
yeah.
pictures here, is this a picture of one growing embryo? Or
um,
are these different embryos at different stages of development?
I think you can actually take one embryo and do a time-lapse image.
Hmm.
And so then you're able to visualize it over time. So like you can place it
So
in
that is just one embryo, developing embryo?
yeah, you can do it that way. Yes, just
Oh,
one
okay.
embryo and monitor it like as an in vivo model over
Mhm.
time. But in some cases, like yeah, you can sort of also do it like the way you mentioned it, like have one embryo at an early stage, then take another image of a different embryo at a much older stage and much aged. So yeah, that's also a way to
Yeah, but it will be more meaningful to monitor one embryo at various stages of growth, right? Because you
yeah,
can see what
changes.
is what remains normal and what remains to be what becomes abnormal in the process. So you
yeah.
can easily detect, and your monitoring would reveal you some kind of understanding that
Yeah, it's like a really tracing like a
yeah.
cell and how it really kind of follows, the events of development. Yeah, those are exciting data that I also kind of
So it's like there is it is like a one form of genetic manipulation, correct? When you am
yeah,
I correct, or correct
yeah,
me if I'm wrong?
yeah. It is
I'm just
like
asking.
it is like technically, yeah, like I am introducing some sort of like random gene into like a specific chromosome. Um, yeah.
Now, I am curious about your experiment, Doc Rommel. Is that expensive? I mean,
Yeah, it's super.
do you need, sophisticated instrument to do that? Of course, fluorescent microscope is very expensive, right?
A microscope is expensive. I think that's what part of my application as a like scientist. So yes, there's the fluorescent microscope, which is very expensive, but the molecular tools,
Hmm?
I
Not
would say not as much because yeah, the molecular reagents, those are pretty, I think, yeah,
They have become cheaper because they are
yeah,
now more popularly
yeah.
and commonly used. At the very beginning, I started off with highly expensive ones. No, I started working with
Mm.
all of those pipetors
Oh
and
yeah,
things like
those are
that. Everything has been expensive, but now it has become cheaper.
mm.
So basically,
Yeah, that's actually one of my plans as a like scientist. So yeah, one of them is this like molecular technique, but I think as I was writing my proposal, I sort of kind of like got the how do I say this? Like I sort of realized the reality of like the Philippines that like some of these tools are pretty expensive and it will sort of wipe out your budget. And so
mhm.
like for a fluorescent microscope, I think yeah, especially the commercial fluorescent microscopes, those are very expensive. And I know I'm kind of talking in very general terms, but yeah, I just like felt like, oh, if I wanted to do this experiment and have like a fluorescent microscope, like yeah, it would wipe out my budget. And so one of the things that I wanted to kind of alleviate that or find an alternative solution was also to create like my own fluorescent microscope. And so this is with a collaborator with the National Institute of the Philippines, Dr. Nat Hermosa. And yeah, we've been really talking a lot about this, like, oh, like this is like the scope that we'll like build. And so with this itself, this would cut the cost by maybe like down to, oh fuck, oh, I mean, sorry, like shoot, I'm trying
50%,
to, about like 50%.
70%.
Yeah, I'm trying to, I think what we saw was this maybe because I'm sorry, I'm having a hard time just because I think of it in US dollars, but then the Filipino peso is like in
no, think about US dollars, so it won't be difficult for you. How much
Yeah.
would be the reduced cost? Because I can identify with you in agriculture, the branded tractors are relatively very expensive, but there are now farmers and agriculturists that have come up with fabricated tractors, and they become cheaper and affordable.
Yeah, so that's the idea, like creating our own like do-it-yourself microscope, and it would probably be like, yeah, maybe 50% cheaper.
50%?
Yeah, more than that actually.
Oh, okay.
So that was like really the goal of this. Yeah.
Okay. So that's another collaborative work, no? Related to your original study about fluorescent microscopes tool for
Yeah, you will have to partner with some engineers and even programmers as well, so that the programming mechanization would be incorporated in that. And we know that we have a lot of, I've seen some here in the University of Philippines in Los Baños, the sound, there are already fabricated sound tracking materials that are being done, and these are invented actually by students together with their professors, so it can be done.
Really creative here.
Yeah.
I'm very impressed by like you know, if you sort of give them the project, the sort of the
but they need the guidance of the biologists, no? In the sound, we attend the meetings of the engineers, but we provide them the basic biological information that to make the proper design. So in your case, go to the College of Engineering and programming or IT and give the instructions of what you want to generate.
You have a very promising fluorescent microscopy in the making here. Which is
it's something that's very affordable. Just something, I know a lot of labs have commercial fluorescent microscopes, but to make it more accessible to like, you know, labs in out there in the provinces where you get access to these fluorescent microscopes, like that's what I hope to sort of build. And we're really still at baseline.
So with your work, how many, I mean, one of the objectives of bringing in Balik scientists is that they have this updated or new designs of technology, information, and etc. And they come home to the Philippines and they have to transfer such information and sets of skills to the local scientists as well as to students. So far, how many are your current proteges, if I may call so, and partners?
Right now it's very small. So yeah, it's just because I've been trying to optimize and troubleshoot everything, and really like the delay has been in part just because the reality of sort of acquiring reagents and equipment in the Philippines just takes a while, just because it's always mostly coming from abroad. And so I've only been able to train few, and it's like one's currently within the lab, so maybe 4 as of now. One is like a DOST scholar, and the other are like MS, PhD students. But the idea is like once I've been able to sort of optimize these tools and have everything ready, then that's when I'll start like doing workshops to begin expanding, you know, the teaching of other scientists. But actually this month I'm, aside from like the transgenics and the fluorescent microscope, we're also doing like this cloning workshop. So the idea is like just being able to learn how to manipulate data, or not DNA, sorry, to manipulate DNA. So to like cut one piece of gene and insert it into another piece of gene. So that's what I'm working on this month. And as of now, we have like 30 people signed up for that workshop. And so I'm pretty excited about it. I think hopefully I can get it out by the end of November, but yeah, probably by early December. That's another.
So you are conducting a workshop on
DNAs.
Yeah,
DNA,
cloning. So the idea is just like
cloning. Okay.
moving one gene to the other plasmid. So
Yeah, maybe you should announce it to the whole UP system so that everyone who wants to come
that's
in.
also being troubleshooted. So that's why we're kind of like, oh, let's try to get this right first. So then we'll sort of, yeah, kind of, yeah.
So that's very good. Oh,
Yeah.
Knows what? If I have no conflict, I might attend your workshop.
I'll give you an invite. I'll like give you a heads up, like, oh, there's this workshop. As of now, it's like internal.
Yeah.
At the Institute of Biology, but we do hope to sort of expand it across. Actually, they're coming to your university, UPLB. I'm not sure.
Yeah.
Nova Lab at some point. They've been doing some workshops over there. Maybe it's within a specific department.
Yeah, maybe it's. I'm sure it would be in my institute, the Biological Science Institute. But you know, maybe they're targeting the molecular biology laboratory or the genetics laboratory. Okay.
Okay, so I think we already more or less highlighted the research of Dr. Romel Santos, and yes, we have reached the time that we need to, wrap up with our discussion on this developmental biology topic.
I can stop sharing.
Yes. So there. So thank you for that sharing. And actually, yes, it's our, how is that? It's our practice, okay, to give a short poem for our resource speakers. And usually it's in Filipino, but yes, I also compose a poem in Filipino, and I will also, perhaps we could also offer it to you, as our token of appreciation, together with my English poem. But considering that our discussion is predominantly in English, so I think I need to recite the English poem that I specifically composed for our guest, Dr. Romel Santos. All right. So the title is the one that you gave me, no? You approved first. Fluorescent Microscopy, Tool to View Brain Development. Is that okay?
Yes, yeah.
Okay. So there. Among living organisms are varied types of reproduction. It can be sexual or asexual in nature. In humans, eggs and sperm will form a union, and from this, a zygote will be formed. Our topic is about developmental biology, science that investigates growth at a stage so early. How are the brain structure developed fully? How does it look? Using fluorescent microscopy. Thus, in today's episode of Kwentong Likas Yaman, Dr. Romel Santos is a balik scientist in UP Diliman. He shared with us his research about development of the brain, for instance, using fluorescent microscopy as the tool to know the images in time. I know that Dr. Romel was born in the US and is not so exposed in using our own language. So I have decided to compose a poem for him in English, which will serve as our token of appreciation for his kindness. Finally, being so young and dedicated to science, I am sure Dr. Romel Santos can go a long way in time. And he can be a role model for our young researchers, making future to be bright for all our budding scientists. Nagkulang na ako sa rhyme. Okay, so thank you for sharing your expertise, Dr. Rommel. We do appreciate your time given to us for this episode of Kwentong Likasyaman. And yes, Dr. Letty, we say something to Dr. Rommel.
Are you there, Dr. Rommel?
Yes, yes, I'm here.
Yeah. Maybe you can leave us with some nice message for the general public and more so for students who might be interested in doing or following your track of research and interests. Give them a bright perspective, a bright future, and the significance of what you are doing.
So yeah, like I think to really start broadly, like continuity in science is very important, and it really starts from like us senior scientists, but then being able to pass that down into like the next generation to take on those responsibilities in science. And it's just very apparent how higher education, especially that emphasizes like science, engineering, math, or anything in the STEM field, is very important because that's what actually produces, the scientists and engineers that the country desperately needs here in the Philippines. And so I just want to stress that really like continue your education and be the engineer or the scientist that you are, and really help this country develop not only in the sciences but like economically, like the types of work that you do helps the economy, and hopefully at some point in the future 80 years from now that our country will hit that threshold where scientists have good salaries and that the job security is very lucrative here. And then, that's what I sort of imagined for Filipinos, that, that we'll come here to the Philippines to do the kind of science that we want to do. But it really starts with, again, continuity, that us senior scientists are able to pass on our knowledge down to these, young generation, and that that in itself, like 80 years from now, they also continue that, passing on the torch, so that just to strengthen this country. And I really solely believe if we sort of play this, long-term game, that eventually that, we'll break this threshold and we'll become, this country will become that, what we hope it will be for us, scientists and everything. So I'll just end it there.
Yeah. So thank you very much, Dr. Rommel, and that's also very inspiring, because the country really needs a lot of scientists to, for the promise of a beautiful future, or a very progressive future, I must say. So thank you for a very engaging discussion on developmental biology. And yes, Dr. Letty.
Yeah, I would believe in what you said, Dr. Rommel, the continuity. And in my age now, I'm senior. I've been in the academy for something like 40 years, and I have seen a lot of progress in the number of students who became interested. And I kind of take pride in the very fact that when like for myself, I started as one single herpetologist. I mean, there was a herpetologist, well-known, Dr. Alcala, and he was Stanford trained and everything. But there was not much yet. But this time, there are more women in herpetology, more, very talented wildlife biologists working out there, being distributed all over the country. I do the encouragement generously, as long as they go and study in this, difficult courses, seemingly not so interesting courses. But once we who are in the position of having understood the needs as well as the challenges and the possible future, we get to inspire them. Like what you have said now, there are very few developmental biologists in the country. But mind you, maybe in the next 3 years, we'll be having more than 500 or 1,000 developmental biologists, that would be a great achievement for us, right? To inspire, to train, and to share. And the students would be to learn, and apply, and to serve. Once they learn, they should not leave the country.
Serve the country. I think that is the
Really,
thing.
yes, yeah.
Okay? And life will be more meaningful. Okay, so I think with that we are wrapping up our episode for today. And again, thank you very much. And for our listeners and followers, we'll see you again for our next episode of Kwentong Likas Yaman.
Please
Bye-bye.
like, subscribe, and share our Kwentong Likas Yaman to all of your friends, and even not all your friends, all those beyond your friendships. Okay, so thank you very much, and God bless everyone.
Thank you. Bye-bye.
Yeah, bye-bye.